And we're going to jump back pretty far, and we are going to talk about this concept of homologous chromosomes. Okay, so homologous chromosomes are chromosomes that code for the same type of information, but not necessarily the same information. So here's a homologous chromosome, and here's a homologous chromosome. Both these chromosomes are going to be located within the same cell. One of them was inherited from the mother, and one of them was inherited from the father. That means that one of them was inside the sperm, and one of them was inside the egg. We're just going to say gender stereotypes here, that this blue one was from the sperm, and the red one was from the egg. Okay? So, we're going to look specifically at one part of them. So we're going to say that this band down here corresponds to a gene for hair color. So the gene is going to be hair color. All right. And we're going to say that the father, the person who this chromosome came from, let's say the father had brown hair. So we would say that the allele would be brown. And we're going to say that the mother had blonde hair, so the allele, in this case, would be blonde. Okay? Following so far? So they're the same gene, therefore they're located on the same place on the chromosome, but they're different alleles, and so they can't be located on the same chromosome, right? So there's a brown allele here and a brown allele here, brown allele here, brown here. Blonde allele here, blonde allele here, right? Make sense so far? I lose anybody? Okay, so what we're going to do is zoom in on this specific area right here. And it just so happens that this area represents a sequence of DNA. So in this area here, you've got, on this side, a double helix of DNA. And on this side, you've got a double helix of DNA. That corresponds to this specific gene. Okay? We're going to magnify it even more. We're going to look even closer at this. Okay, so we're going to take this DNA here, and we're going to bust it open. Okay? And when we bust it open, we're going to do so like this. So we've got, here's this. Something like that. Okay, and then this one is like this. Whoa. Okay, so we've opened up this particular gene. Let's say that the gene looks like this. It's G A T A C A T A C A T A C T. Okay? So, corresponding on the other side then, there would be the corresponding complementary sequence on the other end. I'm not going to take the time to do that, but just know that there would be, this side would also have the corresponding DNA sequence on it. Okay? And we're going to say that in this case, this GA at the front here represents the promoter region. Okay, so this is the promoter. Okay, and so, and this again is not a great example because the promoter is going to be longer than that as far as nucleotide sequence goes, but we don't want to draw like a massive nucleotide sequence here because that would take a really long time. So we're just going to pretend that this GA that's at the front is the promoter. Okay, so what's the promoter good for? What does it do? Yes, the RNA polymerase attaches right here. So this thing is RNA polymerase. And RNA polymerase is going to move down this side here, and it's going to create a sequence of RNA. So here is my sequence of RNA. Let's do it in gray or something. So the RNA comes out of there, right? And that is mRNA. Okay, and it's going to make a chain of mRNA as it moves down there. That's the whole job of RNA polymerase, right? And so then we have this chain of mRNA then down here. And my chain of mRNA has some specific nucleotides on it. And we can figure out what the nucleotides are going to be. So GA was the promoter. And so then we can figure out that the nucleotides are going to be TAC, whatever the complement for TAC is. So T goes to A goes to U, C goes to G, right? That's that. Then we got ATA, so that's going to correspond to UAU. Then we got ACA, that's going to correspond to UGU. TAA, oops, I have one extra thing in there, so we're going to take out one of these A's. TAA is going to correspond to AUU. And then ACT is going to correspond to UGA. Right? And then what's the end of this mRNA going to be? AAA. Right? That's the poly A tail. All right? So we've got our G cap right here at the end. We've got AUG, that's going to be a start codon. Right? So here's AUG, which is a start codon. Okay? UAU is going to be a codon. UGU is a codon. AUU is a codon. And UGA is a codon. Okay? Does everybody agree with me so far? Okay. So, AUG starts us out, and we know that when the ribosome attaches here, so here's a ribosome. Our ribosome has those two different parts. Here's the A site and the P site. Right, this is a ribosome. It's attached to there. Okay. It's going to go and it's going to make the sequence of amino acids, and we're going to start with AUG. AUG corresponds to a start codon, which is methionine, right? You can see it right here, right? So methionine is our first one, M-E-T. Then UAU. UAU is tyrosine, right up here. Then AUU. AUU is isoleucine. So I-L-E. Okay. Then UG. Wait, did I skip one? 1, 2, 3. Which one did I skip? AUG, did I do UAU? UAU, tyrosine. Oh, I skipped UGU. What's UGU? UGU is cysteine. So there's a cysteine in here. And then this was isoleucine. Okay. And then we get to the end here, which is UGA, to this one. So we got 1, 2, 3, 4. And then we're at UGA, and so we go to UGA, and UGA you can see is a stop codon right here. Everybody see that? Okay. So this is going to be stop. So we've now made this polypeptide that it's got methionine, then tyrosine, then cysteine, then isoleucine. Now, is this a super good example? No, because it's only got 4 amino acids in it, and we know that proteins are going to be significantly longer than 4 amino acids. So we're going to pretend that this one is longer than 4 so that it has a nice three-dimensional shape. But let's say that then this thing forms a protein, right? So here's your protein. And this protein is going to look like, I don't know, like this. How does it do that? How does it look like this? What happens to this sequence here to make it look like this? What do proteins do? They fold, right? So they've got like their primary structure, that's the sequence, and then there's the secondary structure, which is the beta pleated sheet and the alpha helix, right? And then they've got tertiary structure, which is the folding, the hydrophobic and hydrophilic interactions. Okay, so this protein is folded like this. Okay, let's say that the whole purpose of this protein is to be a brown pigment. So the specific way that this protein gets folded makes it absorb light, right? And it absorbs light in a way that makes it reflect just a little bit of each of the wavelengths. And if you reflect just a little bit of each of the wavelengths, you end up with something that looks brown. Okay? So we're going to go all the way back up to the top here. There was an allele on this chromosome that corresponded to brown hair. The reason why it corresponded to brown hair is because this allele was a sequence of DNA. That DNA got transcribed into mRNA. That mRNA got translated into a protein, and that protein then was the pigment that made the hair brown in the first place. Right? Make sense? Okay. Let's look at the gene from mom here. This is an allele for blonde hair. Okay? So, what's the difference between the allele for brown hair and the allele for blonde hair? Okay, that's one difference, but let's not worry about dominant and recessive. What's on a more molecular level? What's the difference? What? The sequence of nucleotides. So in this one, we had GATAC, right? And then that was, GA was the promoter, TAC was the first one. ATA, let's change it here to GTA, right? So we just changed one amino acid, or one nucleotide rather. We changed this A over here to a G. We're going to stay exactly the same with everything else. So now we're going to have ACA, we're going to have TAA, and we're going to have ACT. So everything is the same except for this nucleotide. It was ATA on the blue chromosome, and it's GTA on the red chromosome. Okay? So we've got the promoter here. We've got the RNA polymerase that's going to attach to the promoter. The RNA polymerase is going to go down here like this, move downwards. It's going to create mRNA. That mRNA is going to get spliced up. It's going to get its GCAP and its poly A tail added, and it's going to come down here like this. It's going to go out in the cytoplasm, and here is this sequence of mRNA. So we said the only thing that changed was the second codon. So the first codon's the same, it's AUG. The second codon changes from UAU, but now it's going to be CAU. Right? The next one's the same, so it's UGU. The next one's going to be AUU. The next one's going to be UGA, and then a bunch of A's for the poly A tail. Okay? So we've got our ribosome. Here's our start codon, and our ribosome's going to attach, just like it did before. We can divide up our different codons codons. So here's a codon, there's a codon, there's a codon, there's a codon, there's a codon. Okay, and so this one's going to end up with methionine because that one didn't change. CAU is different than UAU, so we got to figure out what CAU is. So we go here. CAU is histidine. Okay, and then cysteine, and then isoleucine, and then the stop codon, because we didn't change anything else. So what changed here? What was the overall change? One amino acid, right? The second amino acid, tyrosine, changed to histidine. You think that's going to have an effect on the shape? Yes. Right? And again, we're talking only about 4 amino acids here. But even if it was 100 amino acids long, changing one amino acid is going to have a massive change in the shape of that protein. So this thing is going to fold to create a protein. This is the folding process. And let's say that the protein that gets created here looks like this. It's really dense up here, and then there's really sparse, and then really dense. Okay? The purpose of this protein was to absorb light. You can see that this one over here has a pretty large surface area that it covers. Does this one have a large surface area? No. So if its purpose is to absorb light, and it has a small surface area, is it going to absorb very much light? No. So we would say that this is like non-functional. Doesn't really work like it's supposed to be. Non-functional protein. It was supposed to do something, which was absorb light, and it is not effective at absorbing light. Okay? So this individual has 2 chromosomes. They've got a blue chromosome and a red chromosome. The blue chromosome produces this protein, and the red chromosome produces this protein. So they're producing both of these proteins. What color is that person's hair? Brown. Why? Because they're still producing this protein, right? They're producing this protein and this protein, but as long as they're producing even a little bit of this protein, that's going to make their hair brown because it's going to absorb light, right? What if they had 2 copies of the blue chromosome and they produced 2 or twice as much of this brown pigment? It'd be darker brown because they're going to produce twice as much brown pigment. That's going to absorb twice as much light, and so their hair will be darker. Darker brown. Make sense? This protein is non-functional. Doesn't really do much. So what if they had 2 copies of this? Is their hair going to absorb very much light? No, it's going to reflect it. What does it look like when hair reflects a bunch of light? Blonde, right? Make sense? So in order to have blonde hair, you need to have 2 copies of the non-functional protein that doesn't absorb light, because that's what gives you the color blonde for hair, right? So this is how we determine whether something is dominant or recessive. Is having 1 copy enough to make you have a certain phenotype, or do you need 2 copies to have that phenotype? Will 1 copy override the other copy? This is a brown pigment, which makes you absorb light. This one doesn't make you absorb light, but this one's still absorbing light, which is going to make your hair brown. What if we're talking brown eyes versus blue eyes? We would have a pigment here that reflected blue light, right? But absorb the rest. And then this one would absorb all of them, right? And so if this pigment reflects blue, but this one absorbs all of them, does it matter that this one's reflecting blue? No, so this would be a person with brown eyes then. See how this works? See how all these things are connected? This is chromosome assortment into transcription, into translation, into protein folding, into protein function. And that's how this genetics thing is all related. Make sense? Okay, now I'm going to throw you a curveball. And that curveball is all of the cells in your body have the exact same DNA in them. Right? Every single one. Your hair cells, your eye cells, your muscle cells, your heart cells, fat cells, all have the same DNA. So what makes them different? If they all can make the same proteins, how is one cell different from another cell? And you have to understand chromosome structure in order to know that. So that's what we're going to do next, is chromosome structure. Okay, and we are going to start really zoomed in, and we're going to get less zoomed in. Okay, so really zoomed in. This is called the double helix, right? And you guys know that that is the shape that DNA takes, right? And if you measured the width of that double helix, it would be roughly 2 nanometers. Okay, it's called the DNA double helix. If you need context for 2 nanometers, they say that your fingernail grows roughly 1 nanometer every second, and therefore 2 nanometers is the distance that your fingernail would grow in 2 seconds. Very small, right? Okay. So, the second way that this DNA is stored, if we stepped back a little bit, if we zoomed out, we would see that there are these proteins. Okay, and these proteins come together in complexes of 8 different proteins. Each of the proteins is called a histone, and they're going to join together into something called a histone complex, and it works like a spool. It's going to take this DNA, and it's going to wrap that DNA around it, like this. Okay, like a spool. These things are going to be roughly 11 nanometers in diameter, so significantly wider than the DNA. And this thing that I've drawn wrapped around it is the DNA double helix, right? So this is the double helix, and we're just zoomed out more so you can't see the helices, but it just looks like one string, okay? This is called a histone complex, or a nucleosome. The third thing is going to be a string of these nucleosomes, and they're going to be evenly spaced out. So here's this one, and this one, and this one, and this one. And the DNA is going to be sort of spaced out like this. Alternating sides. And so when it alternates sides like this, that increases the diameter to roughly 30 nanometers. And they call this the string of beads. It's called the string of beads because when you look at it under a very powerful microscope, it looks like a string of beads. So they call it the string of beads. Okay? Then they take this string of beads and they coil it up. And when I say they, I mean each cell when the cell is trying to condense its chromatin. Right? And so you're going to end up with a coil that looks like this. Okay, and this coil consists of that string of beads. So here's the string of beads, right, on either side. The histone complex is on either side of this coil, and it's coiled up. The string of beads is coiled like so. Okay, the diameter of this is roughly 300 nanometers, so much bigger. Okay, and this is called a DNA coil. Okay, then you're going to take that DNA coil and you're going to coil up the coil. So here's the coil. We're going to coil it up. Okay, this has a diameter of roughly 700 nanometers. And that's called a DNA supercoil. Because it's a coil of a coil. It's actually of a coil. So we're coiled up 3 times now. Okay? And then the last thing, if you had to guess what you were going to do, let's go ahead and coil it up again, right? So we got a coil of a coil of a coil, and we're going to coil it up. And when we do that, it's going to be super tightly wound, like this. What's that thing called? Chromosome, right? And the width of a chromosome is roughly 1,400 nanometers. And that's called a chromosome. Now, the important thing to realize is, when does a chromosome exist? Yeah. Yes, when does that happen? Okay, so yeah, the prophase is when you start seeing the two sister chromatids, right? So it's actually duplicated in S phase, but the chromatids, the time that you can see the chromatids is in prophase. So these only exist in mitosis and meiosis, so we're just going to generalize that and say in M phase. Now, this is important that it only exists in M phase, because this thing is super tightly wound up. And because it's super tightly wound up, you can't actually get to any of the double helix. And if you can't get to the double helix, you can't transcribe it. If you can't transcribe it, you can't translate it. So there's no protein synthesis going on at all when it's all coiled up like this, because it's too tightly packed. You got to undo it to get to the information. Okay, and this is how your cells are going to be different from each other. There's a concept that's called euchromatin versus heterochromatin. Okay, so chromatin is the form that DNA takes when it's not in chromosome form. So every other time, so during interphase, it's in chromatin. And what chromatin looks like is something like this. Here's some tangly stuff, and then here's a nice untangled region, and then a tangly stuff, right? And then untangled region, and then really big tangle, something like this. Okay? So this thing has some really tangled regions here, here, and here, right? And these really tangled up regions are called heterochromatin. Do you think it's easy for RNA polymerase to go into this tangle and find a promoter to attach to? No, it's really hard because it's super tangled up, right? And so these parts are not transcribed, because the RNA polymerase can't get to them. Okay? That is in contrast to these parts that are nicely spread out, and these are called euchromatin. And the euchromatin, it's really easy for the RNA polymerase to get to them, so that part is the part that gets transcribed and expressed. And when I say expressed, we mean made into a protein. Okay? So, within every single cell, there's the exact same strands of DNA. Okay? But, based on the cell type, it's going to have different areas that are all tangled up and different areas that are untangled, and that's going to control which parts get made into proteins and which parts are inaccessible. Okay? So, the concept of going from a stem cell, or a cell that can be anything, to going to a differentiated cell is all about just taking the DNA and winding up parts of it that you aren't going to use anymore. Okay? So, right now, if you're familiar with stem cells are the kinds that can turn into anything, right? And there is a method, a process, where they can take a skin cell, an epithelial cell, and transform it back into a stem cell. And the only thing they have to do in order to do that is get these areas that are heterochromatin, that are really wrapped up, to unwind themselves so that the cell then has access to all of the proteins that it would need to make at any given time. Yeah?
So my heart has different tangled up stuff than my liver, and that's what makes it different?
That is exactly it. Yep. Yep.
Why is it easier to like transform skin cells into stem cells?
It's just that they divide really rapidly. You could do it theoretically with any cell. Yeah.
How do they unwind it?
So we're talking about that tomorrow. It's called acetylation and methylation. We'll do it tomorrow. Okay?